Hi, Lowdown community. I'm so excited today because I've actually been looking forward to an interview like this since I started working on the Lowdown. Today, we get the opportunity to share an update on the first investment that Cancer Fund has invested in. And for those of you that are new to the Lowdown community, Cancer Fund is our sponsor. And our very first investment, which we did at the beginning of this year, is in a company called Reglagene. And it's my pleasure today to speak with Richard Austin, who is the CEO of Reglagene. So Richard, welcome. And if you would, kind of give us a little bit of your background.
Okay, Ann, I'm delighted to be here, and we are so proud to be the first investment of the Cancer Fund. It's a great outfit to be associated with, and it just makes us very happy that decision was taken. So my background, I'm a nerdy scientist. I have a PhD in chemistry, and I went immediately into the pharmaceutical industry, worked for some big companies that you have likely heard of, GlaxoSmithKline and Sanofi, and I always worked on the R&D side of the business. I was a medicinal chemist, one of those people that are designing new medicines and making them, and then really found out that I was more attracted to the business side of the industry. And so about 2010, I began managing R&D operations for Sanofi at its Tucson, Arizona side, kind of a smaller side in big pharma, about 60 scientists. But that really was the key training that helped me prepare for founding a company like Reglagene, understanding what needs to go on the technical side, and then understanding what has to happen on the business, how you actually run one of these things from an operational perspective.
I would think that would have to be beneficial to have both of those experiences when you're starting something up like this. If you don't mind, would you kind of just do a brief recap of what your innovation actually does?
Okay, well, we are creating targeted cancer therapies. And so what our innovation actually does is we are creating medicines that go after specific defects, if I could call them that, in cancer cells. These defects reside on the cancer cell's DNA, and we're able to hit those specifically to cause damage to the cancer cell's DNA, but not damage healthy cell DNA. And the result is that the cancer cells die in a selective fashion. And as you know, we've talked about this, the history of therapies for cancer are just littered with this concept of, you darn near kill a patient to treat them, so, there's a cell killing of cancer cells and healthy cells, and it's really hard on the patient. And even today, most cancer patients start out with those treatments of that type. We're excited about innovations such as immunotherapy and new hormone therapies and the like. But patients don't start there. They start with the classics, for reasons of cost, I don't know, convenience, that type of thing. But we want to be different. We want to create a low-cost therapy that can be used frontline that is just a lot less toxic for those patients.
Excellent. And your medications, is this going to be an oral medication, an infusion?
We are developing medicines that will be administered orally. We see these as being administered in the comfort and safety of a patient's home, but not only that, creating innovations that are not just for the wealthiest people on the planet, wealthiest countries on the planet, but also can be, because of their low cost, can be utilized in developing parts of the world.
Yeah, that was one of the things that I was the most excited to see, is that this would be an innovation that's low cost and available to most of the population. So that's very exciting. Mm-hmm. Now, when we invested, you guys were conducting animal trials. Can you share the results so far?
Absolutely, I can, because you guys invested at just the right time. the Cancer Fund was our last investment in what we call our seed round. The seed round is that first round of dollars that a company like Reglagene will raise to help us really do those proof of concept studies to really validate that the technology works and has a shot to actually create great medicines for patients. And so the capstone of those studies are the animal studies. Now, to be clear, you don't just start there. We actually made 300 drug prototypes to choose the one we wanted to carry into our first animal studies, and the results were just outstanding. We took a very aggressive prostate cancer cell line, one that is actually multi-drug resistant, resistant to virtually all of the prostate cancer drugs that are on the market today. Implanted that into a mouse or into a bunch of mice. You don't do this on just one. You have to have, a bunch of them to see that the effect is real. And we were able to treat these mice for a 57-day trial. Now, for an animal study, that's especially long, especially with an aggressive prostate cancer line like the one we were using. Typically, what will happen with an aggressive line, the cancer cell will figure out a way to defeat the therapy, become resistant, and you'll see the tumor grow rapidly and the mice die. But what we saw is we were able to control that tumor for 57 days with no toxicity to the mice. So no change in weight growth, no change in their organs, nothing, except we controlled the growth of that tumor that we had implanted onto those mice. Now, the mice that received vehicle, that received the placebo, if I could call it that, they were all dead by the end of the study. Because the tumor just grew, and for ethical reasons, you sacrifice them once the tumor gets too large. But we had the vast majority of our treated mice still living at the end of the study, and so we're just incredibly excited about that. From the toxicity perspective, from the tumor growth perspective, and then also too, we've also found, going back to that toxicity point again, is that we've done what are called pharmacokinetic studies to determine how much of the therapy is actually getting into the bloodstream of the mice, and we're getting, virtually 100% of the drug that we're able to place in the stomach of the mice. It gets absorbed into the bloodstream, so it's really efficient at getting into the bloodstream of the mouse. It's there at high concentrations, and no negative side effects were observed in this first trial. Now, I do want to add that these are mice, and cancer's been cured in mice a lot of times, and so you always have to temper your excitement here just a bit. But this was just a great result for us, and what it does is it sets us up to identify the specific medicine that we want to take into what is known as preclinical development, to get ready for human clinical trials, to do those additional tests that will satisfy the FDA that whenever we go into human clinical trials, this medicine will be safe, and that we know how to manufacture it effectively and in a consistent fashion. So that's really the next big milestone for us, is completing those studies and filing what is known as an investigational new drug application with the FDA, so we can begin those phase 1 trials. One more point, if you could let me make it, is that I mentioned it's a targeted DNA damage inducer. So it's causing DNA damage specifically in cancer cells. One of the really hot areas of new cancer drug development are actually therapies that inhibit a cancer cell's ability from repairing DNA damage. So another thing that we have seen and that we've tested is that drug molecules, candidate drug molecules that we have made, whenever we test them in combination with these drugs that inhibit a cancer cell's ability to repair that DNA damage, we're seeing synergy. We're seeing that that the potency of these drugs is better than either one alone, better than the combination. And so these DNA damage response inhibitors, they're actually they tend to be quite non-toxic themselves. And so these should be durable treatments that should do quite well for patients once we're able to make it onto the market.
How big of an issue is treatment resistance?
Treatment resistance, it's truly the greatest problem that cancer patients face today. And we've all lived the story. We have relatives, you know, even I'm sure many people that are watching this interview, the relative or the person has gone to their oncologist and said, hey, I'm here for my checkup. I'm a cancer survivor. And they run the test and they say, I'm sorry, your cancer's back. It's like 70% of the time, that someone has gone into remission, or they've even had a surgery to have a cancer removed, and you say, oh, we got it all, but the thing is that these remissions and these surgeries, it's beyond the limit of detection. We have limits to detection. And so if there's just a little bit left, it can sit there dormant, it can come back later. And whenever it comes back, it almost always comes back more ferocious than what it was before. And like I said, we all know this story. I'm not saying, I would guarantee you everyone listening to this interview knows this story.
Now, you had said that you closed your seed round. What, besides the animal trials, did that enable you to do?
Well, the seed round was really what provided us with the funding to create all these drug prototypes. As you might imagine, when you license a technology out of an academic institution, and the reason why the academic institution doesn't carry this on to commercialization itself is because it's not in their interest. They're interested in starting companies and in licensing their technology, but actually executing on that is not their bag. The professors, they want to write papers, they want to win grants, but developing products and doing all the nitty gritty work, if you will, of developing a product isn't their thing. So once you start to take a technology onto a commercialization trajectory, and it's called translation, once you translate a technology from an academic environment into a commercial environment, you have to find out what's real, what's working, and what's not. And so that was our very first step, was to find out, okay, we've got some great initial data out of the University of Arizona. If you're going to make this into a drug, what do you have to fix? So that's where you identify what you have to fix, and then you start making prototypes. If your listeners and viewers are familiar with what goes on in the startup world in terms of a concept called minimum viable product, this idea that you want to get to a product that you can actually test to see if it works. So what we did in those first several months with that seed money, we made all these prototypes, kept getting better and better and better. And we said, okay, we've got a prototype now that we can actually test in an animal, and let's see if it works. And so we created that minimum viable product. Now we know that it works. So now we got to go to the next level. And it's actually, can we put this into people, and that's the very next thing. And then our next step beyond that is actually putting in that phase 1 human clinical trial. And if I can take a breath and say one more thing, and this is why I'm really so indebted to the Cancer Fund, because small companies like ours, we need groups like the Cancer Fund who get it, who understand that to go from these university environments and to translate that technology into something that a venture capital firm will eventually support, we need people like the Cancer Fund and the Cancer Fund supporters to get us there, because we just frankly don't get there without you. And so some people call this the valley of death. And so we needed the Cancer Fund to get us across that valley of death, to show that this is a viable product and that we can go forward and enable us to speak with these venture capital funders and hopefully get them on board. And
so I know you still have a lot to do. What's next?
What's next? We need to decide on that specific medicine that we want to take into the clinic. So we've got a lot of candidates. We made 300. Making more. We never stopped. We made 300 to get to this first study. We've made another 100 since then, and we're getting better and better and better. And so we've got to choose one, and we've got to do some preliminary studies to make sure it is the one, and then it's officially into those, as I call them, IND and enabling studies to get ready for the FDA. We also likely midsummer into the summer, we need to have that first meeting with the FDA and tell them what we have and how it works and get that guidance for how we can most efficiently get through these IND enabling studies and satisfy their requirements. Now, and as you know, the drug development process is not a quick process. And so our anticipation is that it'll be in the latter half of 2022 before we file that initial new drug application and are ready for that first human clinical trial. But but anyway, like I said, without the cancer fund, we just don't get here. Back to what we are doing. It's a phase 1 trial. We're anticipating about a year for that trial. Phase 2 trial depends on the cancers that are in there. That could be a couple of years. A phase 3 trial, which is a really large trial, longer study, and you want to make absolutely sure that the medicine works and it absolutely is safe. That could be a 2 to 3 year time period there as well. So optimistically, to add all those numbers up to 6 years, where you're maybe 7 years from the market, so it can be a while to get there.
So lastly, is there anything else that you'd like to leave our community with?
I guess from what I know about the Cancer Fund community, I just think that what your community is doing is just so special. I know of no other organization that is really trying to mobilize the people that the Cancer Fund is trying to mobilize to actually make a difference in cancer therapy creation, cancer treatment, outcomes for cancer patients. Normally, it's venture capital firms. Again, they're going after well-heeled people. They're going after institutionals, in institutional investors like retirement funds and things like that. The Cancer Fund is appealing to real people who have experienced cancer in their own lives, people who actually care, people who have written checks to cancer foundations for research, maybe been a little disappointed perhaps that research hasn't turned into products, and really directing them towards technologies and that the Cancer Fund has identified to find the best technologies and bring them to a point to where proof of concept is done and they can be carried forward. So I guess my message for the members of the Cancer Fund is keep going. I mean, we need more people like you. And so I just would ask you to stay involved with the Cancer Fund. Pay attention to Ann. Pay attention to her podcast, because she is very special. And just keep going. Because you're doing the right stuff. You're doing the right thing. And I'm just proud to be among the ranks of the supporters and friends of the Cancer Fund.
Well, thank you for all of that. We're just so excited. And I seriously have looked at, in the past when we were, first doing the Lowdown, I could just visualize the very first time that I was going to be able to do an update. And I'm just so excited. I'm so excited to share this, so excited with your results. It's just, for those of us that know that we have a high recurrence rate, this just provides, as we like to say, more than hope. So thank you so much.
And Anne, if I could just add one more thing before we stop, it is not me. It is the people who work for Reglagene. We have a dedicated team that is running through brick walls and working together to get this done. That is the most gratifying part of my job, is working with people who care. And I'm sure you have that at the Cancer Fund as well. I know you do. I know who you work with. So But that's what makes this ride the best.